Hello, I'm Gail Rodney, Senior Publisher for Neuroscience at Elsevier. Thank you for joining today's webinar. This is part of a series covering various aspects of the publishing process that are especially relevant for researchers. Today, I'm joined by Sam Nastase, currently a researcher scholar at and lecturer at the Princeton Neuroscience Institute, Nick Pullen, Editor-in-Chief of Data and Brief, and Tara Spires-Jones, Director of the Center for Discovery Brain Sciences at the University of Edinburgh, to discuss data management and sharing considerations. We welcome your questions throughout the hour in the Q&A feature. We'll start taking these questions after about 30 minutes, but first I'd like our speakers to briefly introduce themselves. Nick, shall we start with you?
Sure, thank you. So as Gail said, I'm Nick Pullen. I'm a professor of biology at the University of Northern Colorado. It's about an hour north of Denver. I'm an immunologist, so you might be wondering why I'm talking with you, and that's because I'm also the editor-in-chief of Data and Brief, which is where we can publish data articles about any research projects that you're working on. And I will say that I actually started out my career as a neuroscientist. That's my PhD, so I'm glad to be back to SFN some years later to talk with you all and answer any questions you might have.
Yeah, sure. I'll go next. I'm Sam Nastase. I'm a postdoc and lecturer at Princeton, soon to be a assistant professor at the University of Southern California in Los Angeles. I mostly work on human neuroimaging and electrocorticography in the domain of language and communication. But over the years have tried to put in some work in terms of data sharing and building infrastructure and things like that. So I'll try to just unpack that a little bit.
Hi, I'm Tara Spires-Jones. I'm a professor of neurodegeneration at the University of Edinburgh. And like Nick and Sam and many of us, I have many hats. So as well as being a neuroscientist and managing and sharing our own data, I'm editor-in-chief of a journal called Brain Communications and the current president of the British Neuroscience Association. And I'll look forward to sharing a little bit of our strategy in my lab for data management in a few minutes. Thanks.
Thank you. Nick, would you like to share some slides or talk a little bit to your presentation?
Sure, I'm going to go ahead and get this started right now. Okay, so you should be able to see everything I have there. All right, so just a brief presentation about Data in Brief and some issues associated with where to store your data and how to publish a data article. So if you've never heard of Data in Brief, which we call DIB or DIB at Elsevier, it's a multidisciplinary journal, so which means that we cover just about everything. So not just neuroscience, not just basic sciences, but also clinical and applied, as well as arts and humanities. And so we just turned 10 last year and also published our 10,000th article. We actually started as back in 2014 as more of a genome announcements kind of publication. Here's just a little bit of further information about Data in Brief. We publish around 2,300 papers a year. Acceptance rate is around basically 60%. And what you'll see down here, this seems pessimistic rejection rate. I like to think of accepts versus rejects. But what you'll see here is unique to Data in Brief is that a lot of our rejections are actually desk rejections, so rejected by an editor, having to do mostly with formatting or data availability issues, which we can discuss later if you have any questions. We're found in most major databases for publications in case that's a concern for you. And we have worldwide authorship. Most of our authors do, of course, come from the United States in Europe, but we are growing in a few other sectors as well. So something unique about Data in Brief is the type of paper that it is, and it is templated. So there's a template that you can download that we follow to basically describe your data sets. So we have two flavors of submissions that we follow. So we have what's called a direct submission. So let's say you do a research project, you haven't published anything on it, but it's a data set that you want to publish, that you want to be findable out there, to be citable as well. You can do a direct submission and describe it using the template and deposit your data in an approved repository somewhere. We also have what are called co-submissions, which let's say you did that research project, you published it in a journal, so say a field-specific journal, and you have raw data or the broader data set available that wasn't included in that original, parent publication, you can do a co-submission if it's an Elsevier journal that you're coming from with data in brief that says, hey, we've already looked at the analysis of the data, the hypothesis-driven aspect of it. Now here is a description of the whole data set, all of the raw data available in data in brief for folks to reuse and reanalyze whether or not they're associated with that particular study to begin with. So why would we want to be concerned with freely sharing our data and making data articles? Well, part of that is, depending on where you are, you're required to do so now if you're funded by certain grants. So in the United States, if you have an NIH grant, most NIH grants, so for example, over here, research grants, most K awards, or certain K awards, SBIRs and STTRs, you're required to have a data management and sharing policy that discusses how you will make your data available. NSF also has a data sharing policy. And then, of course, if you're in Europe, Plan S is a major driver of sharing data. So not only are we required to do this, but it's also a good thing to do, in my opinion, because it's about open science. Part of it is also, if you're funded through a government grant, it's taxpayer-funded science. And so it should be, in many respects, according to many, freely available for those to reuse and reanalyze, depending on their goals. So data in brief follows FAIR principles, which stands for findable, accessible, interoperable, and reusable. And so we look for this when we're looking at data sets and the description of the data to make sure that it follows these principles. If this is something you haven't heard of before, I encourage you to go to fairsharing.org or to just search these terms about open data and sharing data. And for example, if you go to fairsharing.org, you can find Elsevier's data policy. There's also a data policy specific to data in brief, but really we follow the parent Elsevier policy if you're looking for something. If you find this page, you scroll down and you can find what databases that we support in terms of where to store your data if you want to publish with us. And right now there's a list of 92 approved databases. However, the most common repositories or databases that we see would be things like Mendeley, which is an Elsevier product, which is also part of the Generalist Repository Ecosystem Initiative, which includes these repositories. So we often see people depositing their data in these repositories. So Mendeley, Zenodo are probably the two most common. We also see a lot of GitHub, as well as Figshare and so on and so forth. So I encourage you to look into this if you're looking for a place to put your data. You get a DOI with when you deposit your data in these repositories, and then you can use that to link back to the data in brief paper if you decide to submit. So some other common thoughts and issues real quick that we see on our side when we're looking at data papers. So think about what data format is generally useful and not just what your lab does. So we'll often see things that are specific to a certain lab's practice or a certain institute's practice, and it doesn't really follow fair sharing principles. If you have a new analysis method, I encourage you to write it up. There are methods journals out there. We also have newer paper aspects, for example, perspectives and reviews. So if you have some kind of novel analysis or method around data or data sharing practices, write it up. A big issue that we see in terms of desk rejections have to do with IRB and IACUC compliance, so human subjects and animal subjects. And really this mostly falls on the human subject side, and most people are concerned with sensitive data sets. So for example, electronic medical records. We can't share personally identifiable information openly. However, that doesn't mean you might not be able to have some kind of data paper and at least share some aspects of that. So for example, a couple of years ago, we published a really large data paper describing a really large EMR data set with Cerner. So how do we do that and meet open data guidelines or ideals? We do have in cases like that, you have certain controls that say, hey, you need to be an approved PI who gets access through the people who originally published this paper or through the publisher themselves in order to get access to that data set. Other issues that we run into are large file size, which of course is a concern for neuroimaging. We can talk about later. And then I would just encourage everyone always to pay attention to authorship and what authorship means. because we can run into issues with that. Final thing I'll say is we're seeking new editors and reviewers with expertise in neuroimaging, electrophysiology, computational neuroscience, and/or neurogenetics. So if you're interested, just shoot an email either to dib-me@elsevier.com or to me. And next, I think we have Sam, who's going to talk with you about a very specific kind, to you all, kind of data format called BIDS. Thank you.
Okay. Yeah, so I think what I'll, instead of going too much into detail about bids, what I'll try to do within the next couple minutes is really just, I think bids is a really exciting kind of successful example of this, and I'll try to give a little bit of a motivational sort of idea of like what it takes, in my opinion, to be kind of successful in this. In creating like open standards and things that allow us to actually, successfully share our data with each other. First, I want to just point out some important considerations about data management and data sharing. And this is really inspired by a commentary by Yara Kalchenko and Michael Henke. But if there's one thing that you take home from my little presentation here, I would say that the most important thing when it comes to sort of provisioning yourself to share data is to start as early as possible in your pipeline. So Nick mentioned things like, consent and IRB and so forth, right? Like that needs to happen. You need to figure that out before you even start collecting data. So as early as possible in the pipeline. Ideally, I would love to see people sort of standardize their data, make it ready to share before they even start analyzing their data. I understand that can be difficult in a lot of cases, but you don't want to get yourself into this situation where you've fully analyzed your data, you're writing up the paper. Now you need to go back and convert the data to some standard format so that you can share it. And now none of the analyses that you ran were actually on this new format of the data and so forth. And one thing that I deal with a lot is I really want to encourage people to use very rich metadata with the data that they're collecting, the data that they're about to share, so that the data set can speak for itself. And the alternative to that is that either people just can't use the data because they don't understand it well enough to be able to use it, or like me, you get emails, daily, weekly, asking, like about particular details of the data set. So we try to include as much of that information in the data as possible. And I want to use this as just a moment to advertise the DataLad handbook here. This is an incredibly useful tool here for managing and ultimately sharing data. This is again by Michael Hanke, Adina Wagner, and so forth. But they've put together a really beautiful handbook of how to use this tool. So if you understand the power of like Git and GitHub, this is basically taking that power and applying it to large-scale data. Okay. So I'll talk a minute about BIDS. So BIDS is the brain imaging data structure. This is a standard that has developed in human neuroimaging, primarily fMRI, but it has sort of expanded into other domains. And rather than going too much into the details of BIDS, so BIDS is a sort of standard for organizing your data. I want to talk about why I think it works, because in our field of fMRI, there had been multiple attempts to sort of build standard, sharing languages like this, a vocabulary for sharing your data, that didn't ultimately stick. I think BIDS is going to stick, and I think it's an interesting sort of sociological question to ask why. And I just have a couple points here, right? So one of the things that I think is really important about BIDS is that it builds on existing formats that people are already familiar with, right? So you're not really reinventing the wheel. You're just trying to pull people toward one sort of unified standard. BIDS is incredibly powerful because you're creating a sort of machine-readable file structure with machine-readable data, which means that you can build algorithms that just take only the data set as input and figure out what to do with them, right? This makes automated processing much more effective. And then the thing that I'll just dwell on here for a second is this idea of community-driven development and governance. And this is really the infrastructure, I think, of making something like this work. So I'll go to the BIDS website for a second here. Of course, they have all these, cool examples and whatnot. But the thing that I wanted to point out is this section on collaboration. where they have really rich instructions on how to basically like get involved. They have this whole sort of manifesto of how their governance and leadership structure works, how you can become part of the community, and how all of the decisions are sort of community based, right? They have all of these different working groups, and then they have this really fun sort of process of like extending bids, right? So if you look at these, kind of the extensions that they already have, right? So these are places where people are working on things. So for example, if I go to the Atlas's bids extension, right? This is just a Google Doc, and I can start typing here if I want to and start making suggestions, start getting involved with this process. This will ultimately sort of migrate to GitHub and will ultimately sort of be, merged into the actual standard. So I think developing that kind of community-driven governance structure is probably the most important thing, and why BIDS is succeeding in our domain. And I wanted to just point out, BIDS is highly integrated with OpenNeuro. They are developed by sort of the same kind of community and are working together. OpenNeuro is a repository for sharing. It's very popular for sharing fMRI data. I would say it's the standard for sharing fMRI data, but also can support different data types as well. So I'll close up by just reminding people like who benefits from this process of like data sharing and open science and so forth. And one person is you, right? Like I have benefited my future, in the future, I'm often trying to go back and figure out what I did with a data set or reuse a data set that I had previously collected, right? And having that in a sort of standardized format really makes a difference there. Also makes a difference for just sharing with your collaborators. Really importantly, it allows people who are at, less privileged institutions to have access to data that they wouldn't otherwise have access to, people around the world. And I think it also increases the sort of general public trust in the kind of science that we do, which is increasingly important these days. And so my take home would be make sure to provision your data for sharing from the start, not as an afterthought. And bearing in mind, I know that I'm probably talking to mostly junior people instead of like PIs. This takes extra time. It's a little bit difficult. Your PI probably wants you to kind of hurry along, but I think it's really worth sort of slowing down a little bit and taking the time to kind of do this right. Okay, thank you. So I'll pass it on to Tara next, who is going to talk about a different kind of data format, microscopy. Tara, I'll unshare.
Great. Thank you, Sam. Give me 1 second to get my presentation going for you. Hopefully this will all go smoothly. So I assume you're all seeing that now. So thank you. I'm going to carry on from what Nick and Sam said and just share some of our experiences in the lab about how we manage our own data and some general sort of tips. I'll start off actually with my top tips for data management as a neuroscientist. As Sam mentioned, I'm more of a microscopist than a human brain imager. So we have lots of images and lots of data points. So my top tips are use an appropriate backed up data storage solution. And why do you want to do this? That's because you don't want to lose your data. I mean, you've probably all heard horror stories about people who wrote their thesis on their laptop and the laptop died. But also you have to comply with regulations, right? We have to have our data management plans, like Nick mentioned. In my case, I do use human data, so we have to store our data on servers that are compliant with European regulations because I'm based in the UK, et cetera. And usually your university or your institute will provide a recommendation for where you should store your data. The part that's a little bit trickier for me is organizing and enforcing a folder structure. So I came to this journey, and the reason I was invited to this webinar is I wrote a little paper about this because I was trying to save my future self, like Sam mentioned. When I was trying to go back through our data from people who'd left the lab years before, I realized that their folder structures were all sort of random, and you had to go back through their head to try and remember, organize, find where they put data. So I think it's really important, and I finally just had to make one up because I couldn't find a really good sort of model for this when I Googled it. Another big tip is using electronic lab notebook has been revolutionary for our lab. It's just amazing to be able to link the data to where it is on the experiment. And this makes writing papers easier. It makes sharing data easier. It's great for reproducibility if you can go back to the actual lab notebook where you did the experiment and see the link to the data. I also recommend that we try and archive our data after a given time period. This will sort of vary but based on what your regulations are. Usually funders would want you to keep your data for some number of years, but I'm advocating that we should maybe put it in cold storage after a certain amount of time, because this reduces your carbon footprint and reduces dramatically the amount you're going to spend on this data storage. And finally, I totally agree with Nick and Sam about sharing data for all the reasons that we want to share data, for the responsibility to the public, to the funders, to make the most use of it. But I would say we shouldn't share all the data, and I'll come back to why, because I think we can just do a lot of work for a lot of for very little benefit. So this is our server organization. I went through multiple rounds of trying to organize this, and finally we have gone by date. So we organize things, if we go by talks, which I'm doing now, and we go by year, and then you have to put a certain format. I require a certain file naming format now, because we don't work with standardized data formats like BIDs. We make it up as we go along. We're doing stuff at the cutting edge. We're trying to make new techniques, etc. So we just have to come up with our own systems. But if you're standardized in that system, it becomes much easier. And if you take good notes in your electronic lab notebook, it's even better. So if you have in every person's electronic lab notebook, any experiment they do has their hypothesis. This is linking to our ethical approval, our tissue request, updating the exact protocol that you used, and then definitely putting the location of the data, the location of the images, where they're stored on the server, a link in your lab notebook. And then I'm just going to take this as an excuse to show you some data. So here's an example of what we do. We study synapses. They're colored in white and green here. We study them in Alzheimer's disease. So the big red thing is a neurofibrillary tangle. The study where we published this 3D reconstruction of an image, we shared the analyzed spreadsheets and all the code that we used to write the paper, and that was 127 megabytes. We collated all the raw images, not all the processing steps, because that would triple or quadruple this amount of data, but just the raw images in the study that this image went into was 800 gigabytes. So keep that number in mind for a couple minutes from now. Here's another example. This is a new fun project we're doing with Dr. John Moss at the Roslin Institute, where we're taking tiny bits of brains for people who died with Alzheimer's disease or other forms of dementias. And we're going into excruciatingly beautiful detail with serial block face electron microscopy. And you can see how amazing your brain is. I mean, you're neuroscientists, so forgive me for 1 second of neuroscience instead of data management. This is just showing you the neuropil 30 micron cubed of a human brain. And you can see there's not a single cell body in this. This is all synapses and axons and dendrites, and it's just amazing. And this single image is 70.5 megabytes, the movie that I'm showing you. But the raw data files that make it up are 42.6 gigabytes. And that's just from one tiny bit of brain from one person. So when we want to share our data, it's made much easier by good data management practice. So I know where it all is. It's all collated by date, and I can find it and put it all together. And you can see the image I just showed you is available online. We use our university repository. It gives you a DOI. It's much like what Nick said. It's just free and it's easy for us because it's the one that our university links to, and it's easy to share things off of our university-based server. But have a look at this. The size limit of each entry for our data share is 20 gigabytes, 100 if I beg, and they're nice to me. Right, so think about the sizes of files that I showed you. So what we've ended up doing is a practical thing in our lab is we share all of the spreadsheets, all the numbers that we generate, and all of the code that we use to analyze those numbers. And then we only share the raw images if somebody asks. And nobody has ever asked me. I have shared images with collaborators, but just the amount of time that it would take for us to make everything fair and easy to understand would be phenomenal. And I don't have money to spend on somebody doing that metadata for every single image. So if you want to read more, I wrote about this in my journal Brain Communications, which is an open access journal. It's not Elsevier, it's Oxford University Press publishes it. So you're very welcome to read a little bit more about this if you would like. Thank you to my lab members who generate all the beautiful data and get dressed up every year for our annual party at the UK DRI. And finally, a little plug, as I said, one of my other hats is president of British Neuroscience Association. And if you're able to get to Liverpool next month, we're going to have an excellent meeting where we'll talk about things like this, neuroscience, but also best practices and community. So thank you. I will stop sharing here.
Thank you so much. Thank you so much. Now we'll get to some of the questions. We'll take questions, continue to take questions in the Q&A box. The first question I'll pose to the panel is, and anyone can take this question, or multiple people can take this question, is how do you decide what data to include or exclude with your paper?
I think Tara gave us a really good example of what not to include. So one of the major challenges, excuse me, that I have seen over years of being an editor for a data journal, it's always imaging is the problem, because the files are just so huge. And so what we see there about what to include is exactly what Professor Spires-Jones was saying, is basically share your spreadsheet that has your first pass analysis is I guess how I take it. The other thing that I'll say that this reminded me of, and going back to one of my final comments about the type of data. So in my realm, probably a very commonly used type of data is flow cytometry data. And so that involves a very specific file type, so .csv or .fcs files. You can convert that to a .csv, but what we would look for is make sure you're sharing the data that actually came off the instrument you recorded those data from. And so that would be a .fcs file. So that's somewhat of my limited perspective on things. I'll let if anybody else has anything to say.
I can add. I mean, I think I agree with the point that Tara made here. I would probably, I think in fMRI, we often veer toward the more like maximalist approach of sharing everything, which is probably inefficient in a lot of ways, but we are accelerated in doing that because we have these kind of standardized formats. The one point that I wanted to make is I think it's really easy to share data in a way that like kind of meets the standard of sharing data, but no one will ever be able to figure out what to do with it or how to use it, right? And so it's and this is funny because sometimes I review papers for a journal like Data and Brief, right, where the authors are trying to share their data, but you know, even as an author, you have this kind of blind spot of like you're familiar with the structure of everything and the kind of how the experiment was set up, right? And you just have things in your brain that other people picking up this data set from scratch wouldn't have, right? So I think just trying to figure out ways to, and that's not necessarily the big stuff. That's like the more just like the metadata and the structure, what was happening at what times in this experiment or whatever, that would allow someone to actually kind of reanalyze your data in the same or in a new way. And I think like sharing code, which is light, is also a really useful, important part of that because a lot of the time, just looking at data, I don't know what to do. So I look at somebody's code and I see how they're using the data, and that's how I figure out the structure of the data.
Thank you. We have a question. Is there a particular electronic notebook that you would recommend? That's one of the questions we received.
Sure. I am using one called LabStep, and that's because my funder pays for it essentially, so it's free for our lab. And before that, we used one called ECAT, which is now called RSpace. And I used that because the University of Edinburgh, some of my colleagues generated it. And I've had colleagues who've used things like LabGuru, and I think everybody gets on with the one that they use. And really the important thing is that you are able to track everything. And as a PI, it's amazing. My top tip for starting a new lab is don't start it without an electronic lab notebook because you go all of a sudden from understanding everything that you've done in great detail because you did it to being responsible for lots of other people's work and being able to go in and see day by day what they've done is phenomenal. And I haven't heard any hugely bad reviews of any of the electronic lab notebooks that any of my colleagues have used. So I think the most important thing is just get one. It doesn't really matter which one.
Thank you. Another question. How raw is the raw data and where do you draw that line? Any of the speakers may take that or multiple.
I can start on that one again. So for us, a lot of our raw data is images that come off of a microscope. And even so, when we take the images, we often have to take a whole bunch of images, like you saw the movie going through. So the raw data, we have I can't remember how many images in that stack, but loads. And that would be in a format that's proprietary sometimes to the microscope. So when we share, we don't actually often share the raw data because most people would have to pay to even open those files. We would tend to convert them. I know Nick was saying don't convert it into CSV, but we would convert into a TIFF or something. And the disadvantage is you lose the metadata that came off the machine, but the advantage is people can actually open it. So we keep all of those versions, but in the raw version is the stuff that came off the microscope. And we keep that as our sort of gold standard raw data. But when we share it, we tend to share things that are accessible to more people.
Yeah, and this reminds me, going back to looking at fairsharing.org and looking at, for example, Elsevier's approved repositories. That's just Elsevier journals, but it doesn't have to be just Elsevier. One reason we follow that as a generalist journal is because this is going to be field specific as well. And how, as Sam said, in fMRI, maybe a more maximalist approach is taken there. I think probably with a lot of immunology and the immunology space, a more maximalist approach. But again, we're not as limited. I love the numbers that Tara shared with us. We're not as limited by file size as someone who's doing hardcore neuroimaging or recording videos of some sort. So one thing that those repositories do is they're often society sponsored, or the ones that I just threw up, they're kind of lightly approved by the NIH as go-tos. But especially society sponsored ones have developed kind of standards around how raw is too raw. BIDS is a framework for certain forms of neuroimaging. It tells you, it kind of helps guide you in that to some extent. But again, it can be very field specific.
Yeah, just to follow up on that, I think um, we BIDS encourages folks to share relatively raw data. That said, there are kind of restrictions on that, right? So we collect a pretty high resolution images of people's heads, right? So we have a particular algorithm that we need to run to kind of remove the facial features from that so that you couldn't sort of identify the person, right? So there's a lot of, you can't go too raw because you might end up with sort of identifiable features, right? I wanted to just segue that a little bit because I think one of the things that's interesting about fMRI, at least, is most people also don't want to pre-process raw data on their own. Like a lot of the use cases we find are actually people who would love like a already processed version of the data, right? And so, or some kind of like downstream data derivatives that they can more easily slot into the part of the analysis that they actually care about. And that's also really challenging, right? So kind of going in the other direction, it's easier to set up a sort of standard for like relatively low level raw data, right? But then as you go into more derivatives, different kinds of analyses and trying to figure out what the standard will be for representing those. I would say that's more the Wild West still, but folks in the BIDS community are trying to develop these kind of structures for different standard derivatives of, downstream analyses.
Thank you. Another question is, what repositories do you use the most? I know we've talked about BIDS, and why do they work well for your data?
I think from my perspective, the ones that I see the most would be Mendeley and Zenodo. And then we do occasionally get GitHub submissions. And the reason for that is because there's code usually associated in a description of how to run that code on those data.
Yeah, even in a single lab, we use multiple sources. So I told you about Edinburgh Data Share just because that is easiest and most supported and compliant with all of our regulations. But for our structured data, like proteomics data or RNA-seq data, we use structured repositories like PRIDE. For our code, we use GitHub because that's the standard for code. So we use at least 3 just in our one neuroscience group. And I think it's really not a case of one size fits all, right? It's what's the standard in the community, because some parts, like you were saying, Nick and Sam, some types of data are very structured, right? So we want to be part of that community and use all the backlog work that's already been done. So we put our structured data in a structured repository, which means it has a format like BIDS that we upload it in. And we put our unstructured data, which is just all of our pictures that we take, and then we put that on our institutional repository and we put our code on GitHub. So I think it depends on what kind of data.
Yeah, just want to emphasize that there's different sort of solutions for different sort of fits and sizes and things like that. I would say for almost all of our projects, what we end up doing is like for this project, there's going to be a paper, but we're going to create a GitHub repo that has the code and has some sort of little, tutorial steps so that people can understand how the code actually works. And that is ideally going to be linked to the data that are stored somewhere, right? And it could be like Zenodo, yes, or if it's fMRI data, it could be OpenNeuro. But I would encourage people to build their little GitHub repo in such a way that you can easily like pull down some example data and run it through your pipeline or something like that so that other people can kind of like interact with it if possible.
Thank you. We have a question, and I'm not sure, but I'll pose it. I'm not sure if the speakers will be able to speak to this, but what kinds of safeguards are available in various repositories to prevent your data from being corrupted, either by technical problem or even deliberately by a motivated bad actor?
Yeah.
Is that something we could
Yeah,
answer?
I saw that and I've been thinking about it. You could take the pure pessimistic approach to that and say there's nothing you can do to prevent a bad actor. Or you could actually look at what is being used commonly in a community, what is the common practice within your community. And where I would start is looking at things that are GREI approved repositories. So that's the Generalist Repository Ecosystem Initiative. So those are things like Zenodo, the Open Science Framework, Mendeley, Dataverse, Dryad, Vivly, so on and so forth. And part of the reason I look to those is because those are now, again, they're not technically endorsed by the NIH, but they have that soft stamp of approval because they have stable storage of those data. You get a DOI that's static, at least as far as we have seen for the last couple of decades of the internet. Who knows where we'll go in the next couple of decades. So there's with any venture in life, there's inherent risk. Now, if you're thinking about a bad actor in terms of using your data to do something that either you don't approve of or maybe might be criminal, I don't know if that's where that was going. The latter part of that, the criminal aspect of that, might get to how raw raw data and what data are too sensitive or not too sensitive to share. And so depending on where you live and also depending on things like the Helsinki Declaration, there are pretty well laid out guidelines as to what you can and cannot share. So for example, you cannot share anything that's personally identifiable in an open kind of framework that we've kind of been talking about here. You have to have some kind of control over that. And so those are data that are not shareable. I don't know if I answered it completely, but thinking about a bad actor, I guess it's my inherent trust. I have never experienced an issue where there's been someone running one of these repositories who's intentionally done something nefarious with someone's data.
Thank you.
I can add a little oblique thing to that, right? Which is just a sort of sociological thing, right? Which is that it takes a considerable amount of money and people power to like maintain data and build infrastructure and maintain code and things like that. So like when we think of Open Neuro, it's just like there's just like a handful of people running that. That's their whole job, and I know those people, right? It's like and it's important to keep that in mind. Because again, I know I'm probably not talking to like the executives of funding bodies or whatever, something like this, right? But I think those kinds of roles in science are still dramatically undervalued and under supported.
Thank you. Another question from the audience member. Does Microsoft Team count as an electronic notebook?
I would say probably not. I mean, you could use it in that way. You could use it to keep track of things. But one of the things that the electronic lab notebooks do is they sort of have versions, they have things you can lock, they date and time who did the experiment and where the data are. So I think you could use even just Microsoft Word. You could just keep your own record electronically, and that would be a version of an electronic lab notebook. But the commercially available ones do have some advantages. And if you're in an industry in neuroscience, they'll probably have their own even more strict electronic lab notebook that timestamps things and keeps sort of really solid records for you. If you're doing something like a clinical trial, that becomes very important, right? That you have appropriately locked data and appropriately logged experiments. So I would encourage you to think about, I think there are free versions of electronic lab notebooks that'll be a little bit better than trying to rely on Teams or Word. You could explore. But yeah, I think there are some advantages to the ones that are specially set up as ELNs.
Yeah, and kind of related to that, I just, my perspective as well is part of this is driven by, what your institution is what the culture at the institution is. And so a lot of the work that we do here where I am, and this is not an endorsement for the vendor necessarily, is totally Microsoft and SharePoint driven. And so a lot of the structure that we've seen that Tara shared is kind of similar to the structure that we see in different labs and then departments like academic departments at the university here in a SharePoint kind of environment.
Thank you. What questions do you recommend a lab thinking about developing an organizational system?
Sorry, could you repeat the question, Gail? I didn't catch it.
Sure. Sorry. What questions do you recommend a lab think about in developing an organizational system?
Yeah, that's a really good question. That's what we were struggling with when I had finally decided we needed to reorganize our data. So you need to think about what kind of data you have, how much of it do you have, plan out for the future, how much data are you going to be anticipating per year? Because as a few people have pointed out in the questions, we have to pay for our data storage largely. And think about the sustainability of this. You're taking energy as well as money and time to store these data. So I think starting out with what kind of data you have is the most important question, and then how you're going to organize it. Some people organize their data by project. And so we thought about that and we thought we'll put each grant or each project into its own folder. The problem with that I find is I don't know if other people are more organized than I am, but I find that we'll have one grant and then we'll get another one to do something to follow on. So the project doesn't ever end. So organizing by project was really tough for me. That's why we decided we were going to just organize by year and we were going to have raw data, analyzed data, code, and other sort of systems, and they're each going to be done by year. But you can make whatever decision you like about how you organize, but thinking about how you're going to find it. One other, I don't want to blather on, but one other recommendation I would have is think about in 5-10 years time, when you're going to look for that piece of data, what's going to make sense? How are you going to think about finding that bit of data when somebody says, oh, I loved that image that I saw. Can I get those data to reanalyze them? Think about what you would want to go through to find that data.
Thanks. Anyone wants to add anything to that?
Yeah, I would think about, so I had said before, right, that it's best to kind of intervene as early as possible, right? And the thing that I think about there, right, is that generally, and Tara had said this as well, right? Like different people will come up with whatever structure makes most sense to them. And so I've had to go back and standardize experiments that have been collected and analyzed in a non-standard way. And it's hilarious because even like different people will have completely idiosyncratic ideas as to what makes sense to them in the moment. Their next project will be differently organized than their previous project and things like that, right? And the thing to remember there is you have to sort of come up with some organizational thing anyway. So instead of doing an idiosyncratic thing, try to adopt some kind of standard, right? And so of course, in our field, we already have some standards, so we try to pull those in so that we're kind of already as close to the rest of the community as possible. But then the other thing that I would consider is building, even just locally within a lab. This is kind of, I think, exactly what Tara is describing here, right? But building some kind of like little handbook or something like that locally within the lab that outlines exactly how we do all these things, right? Like how do we get the data from the scanner? How do we organize it as it's coming from the scanner onto our lab server or whatever it may be, right? So that you can and try to build a culture of keeping that sort of up to date and like onboarding people into that process, right? Like that's the sort of thing that we still struggle to do. It's very difficult to do in some sense, but that's the thing that I think we're aiming for, right? Which is that as new graduate students come into the lab, they kind of work through the handbook so that they sort of figure out exactly how they're organizing things. And by the time they're, in their second, third years, they're sort of helping maintain the handbook so that and sort of ushering other people through it and so forth. I think that's the kind of thing that I would try to aim for at least.
And
Thank you.
I just want to reiterate that, from my perspective, it's not that it happens a lot, but it's not uncommon. Try as best as you can to think ahead, particularly if you're working with sensitive patient data, to think ahead about what you can and cannot do with that, because it's really hard to predict the future, but you have to hold yourself 10 years, your 10-year future self to what was our IRB approval? What did the informed consent document say? Because really you're held to that, and it not impossible, but it can be difficult to have some form of de novo analysis at a later date, and you want to try to avoid that was not part of the informed consent, and you want to try to avoid doing that. So a very particular example, not to be vague. Often people will say in their informed consent and their IRB approval, the data are going to be locked up on a hard drive in a locked file cabinet in an office somewhere. That is best practice for a lot of things. But if there's some aspects of those data, like you want to be able to harvest data about some kind of cell type or some kind of protein without any patient identification information, then you can actually structure your informed consent to ahead of time, a priori, to say, hey, anything that has nothing to do with who you are as a person, we're going to hold those data publicly available for the future in perpetuity. And so that gives you that it's in the informed consent, and it gives you that ability to do that later on.
Thank you. There was a mention of costs for storage, and we have a question from the audience. Would like to know if are you aware of any places where they could get free storage for data because they have to pay for it at their university?
So a lot of these places, a lot of the repositories that you mentioned have a minimal, free limit. It's not going to be anything like what Tara was presenting. You're not going to get anything for free with those file sizes. However, one thing I'll mention is that there's this framework called Research for Life. So it's research for the number 4 and then life. And depending on where you are, you may be able to leverage Research for Life guidelines or Research for Life classification to either reduce costs or get nearly free data storage. So what is Research for Life? It's basically a framework that we use on the publication side of things that determines the APC, the article processing charge, whether or not you get a discount or a fee waiver based on resource limitations. So when it comes from country or nation-based resource limitations, would be my best recommendation, is to leverage Research for Life. But it is, I'll be honest, it is a challenge, especially if you have huge data, to find a place to store it economically. I think that's one of the biggest challenges going forward.
I would just add there that you have to be careful with free sites, depending on your type of data. So like Nick was mentioning, we have human data, and so we're required to store those on European servers because of the GDPR, which is this Data Protection Act in Europe. So you do need to be careful. That's why I store all of our data on our university servers. They're backed up and maintained only on European servers, etc. So just make sure that you're compliant with the regulations, which is sometimes hard to do if you are using Dropbox or Amazon or one of these things that will give you a little bit of free storage.
Thank you. We have another question, and what is the best way to deposit Western blot data?
That one's, I've done that many times. Those are usually pretty small images. You can put them in an image file and put them on Mendeley or Zenodo or something along those lines. You might have a field specific or an institution specific place you want to put them, but those are usually pretty straightforward small image files.
And when you publish data, including Western blots, you can use the free storage of the journal, right? So on our journal, Brain Communications, we require full Western blots for any blot you show. If you want to cut it off for the main figure, that's fine, but we require you to upload as supplemental data, the full-length blot. So I would say you're already paying for your publication somehow, so just put it as supplemental data, put all your Western blots when you publish them, and then that's fully backed up and shared if it's open access.
Yeah, that's actually, I totally forgot about that. I mean, I'm unaware of any journal now that doesn't require full blots. And if it's a journal that doesn't require full blots, you should probably find another journal.
Okay. So we're coming up at the top of the hour. Did any of the speakers want to have final words? I'll give an opportunity to have any final words, leave with our audience.
Yeah, I would just encourage, if you're thinking about a new way to analyze existing data or, new data architecture or challenges in data management in your field, that's something that you can write up and you can actually submit to journals. Data Brief is one example. There's other journals out there that focus on methods. There's methods, neuroscience specific methods journals that you could also think about. The reason Professor Tara was invited here was because she had one such publication. And so I'd encourage you to think about that as well.
Thank you. Any other speakers? Any final thoughts?
I was just going to add that I would, another thing that I would consider is before you even start collecting data, see if there already exist open data that you could use. This is like people have tried to estimate like how much money is saved by people reanalyzing open data, relative to collecting new data that they would have to use, rather than collecting the data they would need to kind of do the same thing. And it's like millions and millions of dollars, right? I mean, it's like there's pretty large amounts of money. I'm thinking just in terms of like neuroimaging, right? Where, we have these really kind of standard setting, data sets that a lot of people already use, right? And it depends on the kind of work that you're doing and the kind of questions that you're trying to ask, right? But I think it is useful if you're worried about things like cost. I think it is useful to sort of see if you can reformulate your question in such a way as that you could just use, pose it to pre-existing data rather than having to kind of collect your own data set. And I think that in terms of like, I think some people have a worry that like, oh, this journal is not going to want my paper if it is based on open data, like pre-existing data. That might still be a concern at some really like vanity journals in some sense. But I think my sense is that a lot of journals and still quite good journals, right, are kind of at the stage where that's fine. A lot of people do that, right? That's a sort of accepted practice.
Yeah, that's actually a really good point. And I'll just say, I think the last paper that I published is at the kind of the end cap of that student's work was she had done all this work in the lab and, some animal studies. And then instead of us trying to go out and find a bunch of human beings that we could stab, she actually found a really nice repository that had RNA-seq data for the exact disease state that we were interested in, compared to matched otherwise healthy controls. And it's like, wow, I just saved myself a whole NIH grant. That doesn't mean that, in the future we wouldn't be interested in doing it, but, as a foray into that
Yeah, for like piloting analyses or like for
pilot,
didactic
exactly,
purposes.
yeah.
I mean, there's so many cool things that you can do, with existing data.
Yeah, I'd like to end by saying thank you to all the audience members who came, because you're heroes already. You're generating data, you're thinking about how best to manage it, how best to share it. So thank you. I'll leave you one further anecdote if you need more motivation to organize your data properly. So we have a lab naming system. A big tip that probably wasn't on the slide is structured file naming system, so that when you see a file name, you know what it is, right? So you know which subject it came from and which brain region and which sort of the stains that you have, because all at the end of the day, they're just ones and zeros. So you need to be able to identify it from the file name just in case it gets moved around. And a good anecdote for why you want to do this. So I had some wonderful students who I taught the file naming system. I promise I did, went through it in detail. We have a handbook and we onboard everybody, like you say. But at the end, when they had moved on, these were undergraduates, they were wonderful. They took lots of beautiful images. When they moved on and I was analyzing those images, I had to spend 9 hours renaming all of their files and being a detective, piecing back together, oh, what case could this be? There's a typo. And if you don't have this right spacing, then your code can't parse the, anyway. So just the time that you're going to spend renaming people's files or regenerating data because you can't find it is not worth it. So please do keep going, think about this, come up with your own system that makes sense and stick with it, and enforce people in your lab to stick with it. So that's our weekly lab meeting reminder of please make sure you've named all your files right and you've put them in the right place. And I'm going to check in our weekly meeting that I can find your data. So do enforce it as well.
Thank you so much, Sam, Tara, Nick. Interesting talk. Thank you to the audience for joining us. Thank you to SFN. And have a great rest of the day, everyone. Thank you so much.
Thanks, Gail. Thanks, everybody.